introducing our plenary this morning, who is Dr. Chrissie Painting. It's my pleasure to introduce her because she introduced me just like 2 weeks ago. So Chrissie is a behavioral ecologist who kind of has a bit of an obsession with really ridiculous and underappreciated animals, as long as they don't have a backbone. So she's not really interested in any of those things that have backbones. So she looks at insects with ridiculous weapons and spiders that pretend to be ants, crabs that have claws bigger than their bodies. And she does this really cool job of merging really fundamental evolutionary biology research, but also kind of digging in and doing some of the natural history on these really understudied animals. So she does a really fantastic job of kind of balancing both of those things. Chrissie started her training at Lincoln University and then even did a little bit of a stint working for MPI, or the Ministry of Primary Industries here in New Zealand. She then did her PhD and postdoc with Greg Holwell at the University of Auckland, and bounced around over to work at ANU with Patricia Backwell, who's also a wonderful behavioral ecologist working on fiddler crabs, before coming back to Auckland and then going away to Singapore, to the National University of Singapore, and then coming back to Auckland, where I met her, and she was working with Greg on a Marsden-funded discovery project that she'll probably talk a little bit about today. And then she abandoned me and went to University of Waikato to take a permanent position. I don't know why she would do something like that. So her expertise is really highly esteemed. She's an associate editor for the New Zealand Journal of Zoology and on the editorial board for behavioral ecology. She's also an associate investigator at the Center of Research Excellence Te Punaha Matatini. And she's a working mother to a very verbose and very energetic kiddo. So she's got this really incredible experience navigating the professional world while trying to kind of maintain any kind of semblance of sanity, which I don't envy her right now. And so she's also a great collaborator and a really great mate. So if you're looking for a really fantastic supervisor or someone to work with, don't look any further. She's even managed to get me to work a little bit on insects, which I usually just consider to be a food source for my birds. So please welcome Dr. Chrissie Painting.
Kia ora. Okay, let me share this. All right, are we seeing my PowerPoint?
Looks great, Chris. All
right. So tēnā koutou katoa, no England ko Scotland o tipuna, I te mai o tipuna ki Aotearoa I te tau 1842, ko Mansaroa ko painting o kumatua, I tipu aki au ki Tamaki Makaurau, I noho ana au ki Kirikiriroa. Ki te whare wānanga o Waikato a hau I mahi ana, ko Chrissie Painting toku ingoa, ko tēnei taku mihi ki ngā tangata whena o te rauhi nei, nō reira tēnā koutou katoa. So kia ora everyone, I'm Chrissie Painting, and it's a huge honour to be talking to you today. As Bob actually said in his plenary on Monday, I too, this was the first conference that I attended as a very early and green PhD student a few years ago now, when we all went across to Narrabri in New South Wales, and it was a fantastic time, where I met a lot of the people who I can see up on the screen right now, and who have remained some of my closest friends and collaborators. And so I hope, although we're kind of in this virtual world at the moment, I really hope that the students that are listening in, have been able to make some of those connections and who start to value the kind of amazing collaborative network that we have within ASSAB. So yeah, it's a real pleasure to come and speak to you at my favorite meeting. So I'm just going to slightly minimize this. Okay, cool. So I'm interested really in this sort of central question in evolutionary biology, which is, what drives the incredible diversity that we see among animals in their form and their function? And one of my primary research focuses to date has been on the evolution of behaviours and body structures like weapons and colour and alternative mating tactics that animals use to increase their ability to compete with other animals. And these behaviours and structures are often particularly used by males to allow them to increase their mating opportunities and their ability to father more offspring. And so up on the slide are some of the incredible animals that I've had the privilege to work on over the last few years, some of which Crystal mentioned in her opening address. And they're all males that are up on the slide, and all have varying degrees of craziness in terms of their exaggerated traits. So today I'm just going to tell you a few stories about some of my favourite arthropods and how they've been able to help us understand the drivers of behaviour and appearance variation in animals. So I'll first talk to you a little bit about sexual selection in the wild. I'll then talk to you and focus a little bit on the giraffe weevil for those stories. I'll then talk to you a little bit more in a comparative sense about the evolution of positive allometry and these crazy Brentine weevils. And then I'll touch on some work we've been doing on comparative weapon polymorphism in harvestmen. So firstly, I'll introduce you to the New Zealand giraffe weevil. It's also known as Tuwhaipapa or tüwhaitara, which in Māori refers to its sort of elongated waka or canoe shape. And it's also known as Laslorhynchus barbicornis, which kind of describes the really hairy mustache, that's the technical term, on the underside of the rostrum or the elongation of the head that these males have. Males and females are so very different in appearance that actually back in the 1700s, when Fabricius gave them their Latin name, he actually gave them males and females, two different species names. And these stark morphological differences between males and females is what drew me in to study them in the first place during my PhD. And so they turned out to be a really excellent model for doing some work on sexual selection in the wild. And often a lot of what we've learned about sexual selection in insect species is from work that's been done on a handful of really important key model species in the lab, so species like Drosophila and seed beetles and crickets. And although these studies are really powerful for being able to tease apart selection mechanisms in a controlled setting, they may not always capture the reality of selection and the complexities of the wild. But there's a good reason that people tend to avoid this. Studying insect behavior in the wild can be hugely challenging. Often, species are mating in flight. They might be nocturnal or mate underground or just generally be quite secretive. But it turns out that the giraffe weevil is actually an ideal model for being able to go out and observe them in the wild. And that's because during summer months, both males and females form these really large aggregations on fallen trees. So they all come together on these recently fallen trees, and females will be looking for sites that they lay their eggs, while males will be looking for females to mate with. And so as well as that kind of, logistical aspect of working on them that's turned out to be really beneficial, they also tick the box in terms of all these sorts of crazy components that come together that allow them to be a good model for sexual selection study. So I've already mentioned that they're quite highly sexually dimorphic, and I think this photo kind of nicely illustrates that as well. So this is a male here. You can see my mouse, right? Moving, yeah. So this is a male here standing over a female, and you can see the male has this extremely elongated head. Its mandibles are right up at the very end here, so that's what he uses to bite onto his opponents and try to throw them off trees. Females tend to be a lot smaller, and they use their rostrum to bury into the wood to make these holes in which they'll lay their eggs. So as I've mentioned, males have these really elongated rostra, and they actually use those as weapons. So they use them to try and basically beat up an opponent male that's on the tree to try and gain access to that female to mate with. In addition to that, they're also extremely size variable, and I think that they're one of, if not the most extreme examples of adult size variation in the animal kingdom. So males can be, I'm talking just about adult males here, adult males can be as small as 15 millimeters in body length right through to 90 millimeters, so that's a 6-fold difference. And even in body mass, which we haven't even measured the most extreme on either end of this continuum, but males can be up to 30 times heavier than their smaller counterparts. So the kind of a human equivalent of this is that if you looked at the difference in weevil size from smallest to largest male, it's the equivalent of one human relative to 2 large giraffes combined. So this whopping difference. And so it's not really surprising, I guess, then that we see the evolution of alternative mating tactics. And so when there's a really high disparity in competitive ability among groups within a species, in this case, males of different sizes, then we tend to see the evolution of different behaviors that the disadvantaged group evolved to be able to try and still compete and get some mating success. And so the case of the giraffe weevil, my early work showed that these small males are using sneaking tactics, and I'll show you a video of that a little bit later. In addition to this, kind of more of the female side of the story, is that females are really, really highly polyandrous. So they'll mate many times throughout their short lifetime. So adults only last a few weeks or months, but within this time, females will just basically be mating continuously while they're looking for sites to drill their eggs. And so we know that insects store sperm, and so this kind of leads to all sorts of interesting questions about the interplay between traits that allow males to be really good at just gaining mating opportunities in the first place, and traits that allow them to actually have high fertilization success and through sperm competition. So I'm not going to talk about this stuff today because this is actually work that I'm currently doing with my Rutherford fellowship, but hopefully you'll hear about that in future talks. So how did I actually go about working on the species? So my approach really has been largely based on field observations. As I mentioned, they're really a good species for being able to do this. And so the way I did this during my PhD was that I found aggregations of both males and females during these warm summer months. And I'd go out weekly and mark basically everything that was on those trees so that I got a really nice longitudinal data set that I could do macro capture with. And then because they were all marked and measured, I could do focal animal observations within those aggregations. And there really were large numbers. So over the 3 summers that I worked on them during my PhD alone, I marked almost 7,000 weevils. And this gave me a massive data set for both morphology and behavior. So I'm not going to bore you with heaps of the kind of graphs and stuff from my PhD. Instead, I'll just show you this lovely video of a couple of males fighting. So this is a female here, and she's mating with this male, and this intruding male's come along and trying to biff the big guy who's about the same size off. And you can see he's using his mandibles right at the very tip of his rostrum to try and pull the opponent off. And because we're able to really closely observe these fights in the wild, we're able to describe the different elements of their contests, determine how males are actually deciding when to withdraw from a contest, and kind of apply these game theory models to their fights. And so we're able to figure out that they're likely to be using mutual assessment to kind of figure out when they should decide to leave, like this guy here. And bigger males will win contests 90% of the time. So the rostrum and body size itself is playing a really large role in contest success. I also mentioned the sneaking behavior. And so if you watch this little male here interact with this larger male, and he's mating with a female at present. And so rather than bothering to try and kind of bash up the other male that's there, he just totally pretends he's not even doing anything and moves his way in between the male without really physically interacting with the guarding male at all. And so the sneaking behavior allows small males to gain mating opportunities without relying on having a big weapon to fight off males. You can see now he's actually kind of tucking himself down the side of the female to stop him from being able, the resident male from being able to pull him off. But one other really neat thing about their behavior is that small males will still fight and guard in the same way that large males do, but only when they're opposing really similarly sized small males. So in this way, they actually show really flexible alternative mating tactics that allow them to make a decision on whether to fight or sneak, kind of in the best, in sort of what is the best in that moment of time. So how do we actually go out and measure sexual selection on a wild animal? Well, we go out and we watch them in the bush. And this is some work that Bex LeGrice did during her master's project, where she actually did a really amazing effort where she went out daily and watched big aggregations of weevils, both males and females, and counted how often they were copulating for a 30-day period. And she was basically making the most of the fact that these weevils have relatively high site fidelity, so they'll actually stick around and stay on the same tree for quite some time. And so she was able to basically, by having marked individuals, look at how body size relates to mating success. And what she found was that both bigger males and females will mate more often than smaller males or females. So this line here is for males, and you can see it's a pretty shallow selection slope there, but nevertheless positive. And it's mostly kind of, I guess, driven by the fact that small males often we won't see copulating at all, and larger ones we always do. So that's driving that positive effect there. For females, the result is really interesting as well, because females, their mating success is well, their mating number is mostly to do with males choosing to mate with them. And you can see there's a really strong relationship there that the bigger females are much more polyandrous than smaller females. And that is super interesting for 2 reasons. So first of all, some work that Sarah Hawkins did recently for her master's showed that bigger females are more fecund. So they have both higher number of eggs and also bigger eggs as well. So they probably are quite beneficial for all males to try and mate with. But given that we now also know that these big females are more polyandrous, that kind of poses an interesting trade-off there, but with trying to mate with a female that's perhaps more fecund, but also is potentially storing a lot more sperm from a whole bunch of different competing males than a smaller female. So this is some of the kind of interactions here between pre and post-copulatory traits that we're trying to get at with some of the work we're doing now, where we genotype the eggs and look at which male is actually a successful partner. During Bex's master's, she also analysed 2 years of microcapture data that I'd collected during my PhD, and she actually looked at survival and recapture probabilities for males and females of different sizes. And contrary to what we expected, we actually found that both males and females, the bigger they are, the higher their survival probability. And we actually didn't expect this. We predicted that bigger males may pay for being large by having either higher predation rates or just higher rates of senescence or something like that, but that's not what we found. And so what this kind of came down to is that larger individuals, both males and females, have higher survival rates, but they're also, so they're living longer, but they're also having higher rates of mating success. So for males especially, that means accumulating more matings over their lifetime. So as well as looking at this kind of puzzle of extreme interspecific variation in body and weapon size and how this couples with behaviour, I've been intrigued by why sexually selected traits can vary so much across the geographic range of a species. And so to do this, at least for the giraffe weevils, I've been sampling them across their latitudinal cline. So this is 11 sites along the North Island of New Zealand, spanning about 5 degrees of latitude. And I've measured giraffe weevils at each of these sites. And what I found was that there was a really strong latitudinal cline in body size. So as we move south, so down the country into cooler areas, we find that body size on average increases for both males and females. So these figures these are just the acronyms for the sites, but they're arranged in increasing latitude. So as you get further south, body size is getting bigger for both species. And so that rule kind of follows Bergman's rule in terms of this idea that, at least for endotherms, that there's some kind of mechanism around heat conservation in cooler areas, although it's less clear how that applies to ectotherms. But what I think is really fascinating was that when we actually looked at the relative investment that males are making into their rostrum size, this too varied along the cline, but in the opposite way to what we expected, or at least the opposite way into which body size varied. So the way we looked at the relative investment of that individual made into its rostrum, we calculated the allometric slope for each population. So that's basically saying how much ratio of body to weapon does a population invest into. And so what we found is that across the cline, as we're getting into these cooler areas, males are on average having much shorter rostrums. So their rostrum are actually getting stubbier in these cooler areas, even though their bodies are getting bigger. But when we compared that to females, we found that there was no variation in relative rostrum size across that cline. So basically, rostrum are staying the same ratio to body size for females across that latitudinal cline there. And so we thought that was really interesting because something is obviously going on that's driving this pattern in males, but not for in females. And at the time when we published this, and I actually still am not really aware of any examples where we've seen variation in in relative weapon size across a latitudinal cline. And there's lots of hypotheses for what might be driving this pattern, including that sexual selection and natural selection pressures may vary along the cline, which drives increasing body size on one hand and decreasing investment in the other. And one hypothesis is that given this really strong correlation between latitude and temperature, the temperature itself might be playing a really significant role in driving weapon and body size patterns in the species. So I just want to present a little bit of work that Nicole Haydower did for her master's recently in collaboration with Crystal Kane and Alexei Rushman and also Matt Simons. So Nicole did this really great experiment where she brought weevils into the lab. She mounted them onto a little bit of plastozote so they couldn't move, but they were alive. And she popped them into these 25-degree incubators. And because the ectotherms, they shift their temperature in accordance with whatever the ambient temperature is. So they would rapidly move up to 25 degrees. And then once they were acclimatized, she moved them very quickly across into a cooler incubator. So that was set at 15 degrees. And inside that incubator, she had a thermal imaging camera. And that camera was set to take photos every 15 seconds for 10 minutes. And so what she was able to do from that was get these amazing thermal photos over time. And so this shows how basically the animal was getting colder over time. And she was able to not only relate that then back to body size, but look at how different sections along the body were varying and how fast they cooled. And so this is what she found. So this figure is showing body mass along the x-axis and cooling rate on the y-axis, with increasing values on this y-axis, meaning that the trait cooled faster than down here. And what she found overall was that the rostrum, so that's this line here and this line here, so the tip in the middle, are both losing heat a lot faster than the rest of the body, which are these two areas down here. So the central part of the body, like the really big chunky bit in the middle around the elytra and the abdomen and the thorax, those are losing heat a lot slower than these extremity parts that stick out the end of the head. But interestingly, this is also really body size dependent. So large males are losing heat from those extremity points from the rostrum a lot faster than smaller males, but they're retaining heat in the central part of their body for longer than small males. And so this really closely matches what we're seeing in terms of morphology, where larger males in the south could have shorter rostrum to kind of work some way into maintaining heat exchange. But we still need to work a little bit on those actual proximate mechanisms of how that's actually working. But a pretty neat experiment to kind of support those patterns. So overall, I guess I wanted to just say that I think giraffe weevils are a really excellent model for doing some of these sexual selection studies in the wild. Particularly for trying to kind of tease apart these drivers of the interspecific variation in morphology and behavior. But I'm also really interested in how sexual selection can drive variation and the extent that a species invests in weaponry among species. And one way to do this is to put things into a comparative framework and to look at the evolution of allometry. I've already used that word lots of times, but I should explain a little bit about more what I mean by that. So most traits will scale proportionately with body size, where large individuals are really just scaled up versions of small individuals. And when you plot this onto a log scale of body size against trait size, the slope is equal to 1. So that's this isometric slope here. So Drosophila wings, for example, show isometry. But many traits don't scale proportionally with body size. So for example, genitalia, courtship, mate-searching structures all tend to be negatively allometric, so have this slope of less than 1 on a log scale. And this means that there's not very much variation in the trait in relation to body size. So for example, the little lantern on a firefly is about the same size no matter what the body size of the animal is. Likewise, there's also a really large number of species which possess traits that show increasing investment in the trait relative to body size, and that shows positive allometry and a slope greater than 1. And many sexually selected traits used as weapons show this pattern. And the repeated evolution of these positive allometries across many different species suggests that this relationship between body size and trait size is highly evolvable. And I've been interested in investigating patterns of allometry among these groups of different closely related species and looking at whether the degree of sexual selection pressure can predict the evolution of positive allometry. And the Brentine weevils are a fantastic group to do this in. So this is the same subfamily that the New Zealand giraffe weevil belongs to, but it's one of about 1,700 species that are distributed mostly across the tropics, but a few outliers into temperate areas as well. And some species look just kind of like the giraffe weevil. They're long with these really elongated heads. Each of these photos is a male on the left and a female on the right to show you the differences between the sexes. As well as those elongated forms, there's also a whole bunch of species where males have exaggerated their heads in a different direction. So they're a lot shorter and squatter, but they really broaden and have these giant mandibles sticking out the front. There's these weird ones where they've got kind of small heads, but really massive hind legs with spines all over them, and those are lacking in females. And then there's, unfortunately, also lots and lots of species where there's practically no sexual dimorphism at all, and there's no exaggerated traits. So males and females look practically identical, especially if you're not a taxonomist like me. So what a great group to try and look at these kind of comparative evolution of allometry. So together with Greg Howell and Matt Simons, we've been exploring the relationship between sexual selection pressure and the evolution of positive allometry using these fantastic Brentine weevils as our model group. And this was work I did as a bit of a side project during my PhD, where I visited museums around Europe and took lots and lots of photographs, and ended up with enough that I could build allometries for 59 different species across the subfamily. And so we looked at different traits. So we measured what we thought would traits that would likely be under sexual selection. So it's both different measures of the head. So one is the full length of the head, and the other is this pro-rostrum width here, which looks a little bit silly on the giraffe weevil diagram, but you can imagine in those species which are really broadened, both either in their mandibles or the head width, that this measurement kind of takes into account that kind of exaggeration. And then we also compared these allometries of what we expect might be under sexual selection to a non-sexual trait, which is the elytron length, because it's important to compare both across sexes and also to other traits on the body. So to account for non-independence in our analyses, we did some phylogenetic least squares analyses, so that takes into account phylogenetic relatedness between species. And we use sexual size dimorphism as a proxy for sexual selection pressure because, as you might expect, there's pretty much no behavioural data for pretty much all of these species. And so we're able to use this proxy where basically the idea is the more sexually dimorphic the species is, that it's predicted to be under higher sexual selection pressure. And that's been a proxy that's been used many times for species that have a lot more behavioural data than the Brentines. So we then looked at the relationship between sexual size dimorphism and the allometric slope for both males and females, and for these 3 different traits that we measured. And so on these figures, each little dot represents one of those 59 species. And as predicted, we found that a positive relationship between the steepness of the allometric slope for males and the degree of sexual size dimorphism was there. So basically along this figure here, you've got increasing degree of sexual size dimorphism and steeper allometric slopes here. But we didn't find any relationship for females, which is to be expected. I think there was only one example of positive allometry in female head length. Almost all of them were either isometric or negatively allometric. So not much variation in rostrum size, head length for females. And I'm going to come back to this funny little outlier a little bit later. We also found a really similar relationship for our other measure of exaggerated head size, which is pro-rostrum width, where we've got this really strong relationship with more sexually dimorphic species having more higher rates of positive allometry in males, but again, no relationship in females. Again, most of them were isometric or negatively allometric. Surprisingly, we did find a positive relationship between the degree of sexual size dimorphism and the strength of the allometric slope for males when we looked at elytron length, which we thought was a little bit surprising because we had not predicted that this trait would be under sexual selection. We've not seen it being used during sexual displays or fighting in any of the species that have been worked on. But I guess what's really important is that lots of species that bear these exaggerated traits are often supported by disproportionately enlarged structures that help them to move around. So things like exaggerated wings or legs that compensate for any negative impact on performance that these exaggerated traits might inflict. So while elytra aren't used directly in flight, they do correlate very strongly with the length of hind wings, and that may compensate for the ability of these large individuals to be able to carry these big exaggerated heads during flight. And I told you I'd come back to that funny little outlier. So this study also revealed a couple of really wacky species that showed very interesting within species variation, not just in weapon size, but also in weapon shape. And in this species, males can be one of 2 major male weapon phenotypes. So we can have large-bodied males that have these really elongated heads, so that's this alpha male up here, or you can be a big-bodied male but have exaggeration in the opposite direction. So you can have a much shorter head, but it's really broad and with these kind of frightening-looking mandibles sticking out the front. And this is really fascinating because typically male polymorphism is divided into males of low condition having small reduced weapons compared to males of high condition that have really big exaggerated weapons. But here we have 2 different big-bodied males that have one of 2 different major weapon types. And just by pure coincidence, we happen to be also working on polymorphism in harvestmen at the same time, and we found a really similar pattern within many of the species of neopilionid harvestmen that we have here in Aotearoa, where there's not only minor males and major males with tiny or exaggerated weapons, but also examples where major males could be one of 2 different weapon shapes. So in the case of these harvestmen, and this is the two different species that we've described the polymorphism so far, males can have these really, really long kind of crane-like chelicerae or jaws hanging out the front of their heads, or they can be likewise big-bodied but have much shorter but really broad chelicerae with pretty big chunky pincers on the ends. Or you can be a really small male with tiny little reduced chelicerae. And so likewise, Erin Powell, who did her PhD on this species at the bottom, found a very similar pattern with that species. And we've been using a geometric morphometric approach to explore some of the variation in weapons among males of different sizes and among sexes and species as well. And I really like this image that Erin produced for one of her papers recently, which maps on the actual photos of these chelicerae from both males and females onto this PCA plot, which basically just looks at variation in shape space for each of these individuals. And what we found is that, or what she found, I should say, is that females kind of cluster off down here. And then this group here are the really big-bodied males with these kind of short, broadened chelicerae. And then we get this other big male group over here, which are mostly the really, really long, slender, big-bodied males, but also includes all these small males as well. So it looks like the little ones are just kind of scaled down versions of these really, really long, elongated males. And at this stage, we don't know how these different weapon shape morphs are actually determined during development. One hypothesis that we have is that with both the harvestmen that I've just shown you and also that green-time weevil that I mentioned earlier, is that there's kind of a combination of a facultative conditional threshold and a genetic polymorphism. So the conditional threshold relates to the body size and determines how big the weapon is. So that might say sit along here, and below that threshold you have a very small reduced weapon, and above it you kind of go fully into producing a big weapon, while the genetic polymorphism determines what kind of weapon shape you actually have. So in the case of the harvestman, it might determine whether you have a really long, slender chelicerae or a really short, broad one. So we haven't tested this hypothesis, but I think it's really important to show is that these kinds of species where you're getting really extreme divergence within a species allow you to have these really opportunistic kind of opportunity, that doesn't make any sense, to be able to try and figure out why we get so much divergence within species, and that could help us to explain why we see so much divergence among species too. So we've also been exploring this in 3D as well, using these amazing micro-CT scanning facilities and building these 3D reconstructions of male and female chelicerae. And this is work that's been done in collaboration with Jamie Gray, who was a postdoc in Greg Howell's lab for a short time, and who created these amazing 3D models. So beyond just categorizing males into different morphs, we're also taking this comparative approach to understand weapon evolution in the family. And we have about 100 CT scans now for 15 different species within this group in Aotearoa. And that gives us a really powerful data set that we can apply to all sorts of questions about weapon evolution. And there's some really, really crazy examples of weaponry in this group. My favorite being this one here, Philstripsalis bona, where males can be one of 3 different kinds that we've found so far anyway. And they can have these massive kind of dilated fingers on the ends of their chelicerae, with even 2 different forms of where the actual teeth are found along the chelicerae. And we know that this is the same species because we've been able to look at this within the phylogeny as well. So Gonzalo Giraffe and his team are currently developing molecular phylogeny of the group that we'll be able to use to kind of map on our weapon morphs. And we'll be able to explore patterns of weapon exaggeration and positive allometry. But what we're really excited about is being able to look at the rate of evolution of weapons versus other traits between males and females, and also even within our different morphs as well. So we'll be able to look at predictions that our major morphs, which seem to show a huge amount of variation in this group, have evolved at a much faster rate throughout the phylogeny than our more conserved shapes within females and minor males. So today I've told you a few stories about some pretty amazing New Zealand invertebrates and how we can use these models for increasing our understanding of behavioural and morphological variation in animals. And while the deep body of work that's on more traditional model insect groups have been obviously very hugely important for developing the field of sexual selection, I think it's really important that we're adding these less well-known model groups to our collection of studies in this area. Especially those that we're able to go out into the field and kind of understand how the complexity of these wild situations is relevant to the behaviour and the morphology that we see. So I guess if there was a take-home message from this, I'd say that you should all come to Aotearoa to study sexual selection on some of these really quirky invertebrates. And I also was just asked to talk a little bit about my journey to becoming an academic. So this is me when I was about 2 years old, and funnily enough chose a very similar photo to the one that Bob used in his plenary on Monday. This is me giving one of my new chickens a cuddle. And I grew up in Tamaki Makaurau, or Auckland, in the suburbs, but I did have a little bit of bush around me. And so my brother and I were given a huge amount of freedom to kind of explore and really get into nature. And so while I guess it kind of looks obvious to me now when I look back that maybe I'd end up doing something with animals, it certainly wasn't clear to me at the time. And I really didn't know anything about what being a scientist meant until I was, to be honest, probably halfway through my science degree at university. So I was the first in my family to go to uni. My dad managed a motorbike shop when I was a kid. My mum worked part-time as a cleaner when we were little, and then she eventually joined a market research company when I was a bit older. But in my teens, I had this strong interest in gardening, and I really liked horticulture, so I thought I'd go to uni and kind of learn a bit about how to grow plants, because why not? But when I got there, I realized ecology was obviously for me, and that's how I kind of went down that direction. And Crystal's already mentioned a bit about my academic career, so I'll just quickly put this slide up now. But I guess I wanted to say within this that I've had a fairly linear pathway in my academic career. I've kind of been very lucky to have postdocs that kind of eventually led into my permanent position that I've got now. So as Crystal mentioned, once I returned from my postdoc at NUS in Singapore, I had this longer term Marsden funded project, which is where I did some of that work on the harvestmen. But actually most of that time was taken up by this little fellow who I had in 2017. And amongst all that chaos, I also applied for the job at the University of Waikato, where I am now, and obviously got that job, which was great, and got the Rutherford Foundation postdoc at the same time. So I've been able to take that with me, and it's helped me immensely for being able to have some research funded. So as well as sort of telling you a little bit about where I have come from, I was asked to talk a bit about a personal challenge that I overcame. And so I'd like to talk a little bit about the challenges of returning to work after having a baby and balancing parenthood and having an academic career. Because I guess as scientists, we often want to keep our home lives and particularly our family lives separate from our professional selves. But it's interesting, I've been reflecting a little bit about what Bob mentioned in his plenary on Monday about how what you bring, like thinking about what you bring to your role and how you can bring your whole self to your job. And for me, that's my life as both a scientist and a mum to this crazy little chicken cuddler. And so this photo, right, this makes me look like I've kind of got it all sorted. I've got super mum status down pat. I've got my baby on my back. I'm out doing a bit of field work. But the reality is we're just out for a bushwalk and I found some giraffe weevils that I can't help myself, right? So I don't take my kid into the field. For me, that just would be way too much hard work. I've got sharp tools. He could probably fall off a cliff. So really the reality for me of having a work life kind of balance is this. So the reality for me is I'm usually exhausted. I've got these huge bags under my eyes. I was walking lots of kilometers a day, not to collect data, but just to try and get my baby to sleep. And so, of course, all of these things are still in play when I came back to work, still dealing with these challenges of learning how to be a mum and also how to be a scientist and both of those things together. And I think as a lot of us are A-type personalities that are really used to having it all under control and things going fairly well for us, at least in our careers, parenting is that one thing where it's completely out of your control for most of it, both having the baby and then looking after the baby once it's there. And so I was obviously still pretty exhausted when I came back to work, and I was dealing with a lot of guilt about setting off my kid to be looked after by a stranger, and also then not being very productive at work because I was distracted half the time and was only working a couple of days a week. And on top of this, I also had the logistical challenges of trying to breastfeed and finding time to pump milk while I was still somehow getting some work done. So I wanted to, I guess, talk a little bit about some of the things that helped me just really briefly. But I should acknowledge that I was 100% in a research role as a postdoc. So in some ways, I guess I was lucky in that I wasn't dealing with teaching obligations during this time other than some postgrad supervision. So I had, I guess, fewer things on my desk than if I had a faculty position. The one thing that really helped me returning to work was to have really flexible working hours. So when I came back for just 2 days, I slowly ramped this up to closer to full time as my son got older. And it was really tricky to work out what constitutes being part-time versus full-time, because that's what you're used to doing, right? So having a lot of flexibility around deadlines and lowering expectations about what you can realistically do and on a part-time schedule was really important for lowering some of that guilt that I found around lost productivity. And another thing was, I guess, really important to me was having really supportive colleagues. So particularly my postdoc advisor, Greg Holwell, was incredibly important for making me feel like I could prioritize my son over my work during this really difficult transition back to the office. And I guess I would suggest that it's really important to have these open discussions about your priorities for yourself and your family, as well as what are really important priorities for your job and any deliverables that you might have around that, both before you go on parental leave, but also when you come back. And you can revisit this as much as you need to over time. And this can also be really important during pregnancy, because oftentimes you're just not well enough to be able to go and do field work or lab work. And so you have to have flexibility around that as well. Maybe I'll just mention one or two other little things that really helped for me. So a really big thing was having some on-campus resources. And some of these were just really minor things, like the equity office gave me a little fridge that I could store breast milk in while I was at work, so that it didn't have to be in the tea room and have any awkward situation involving breast milk. And while this was a really small little signal, the thing that it felt to me was that I belonged back at the university as a mum. And so while this didn't really cost very much at all to the university, it certainly was a strong signal to me that I could be there and I could still be part of the community that is the academic science community. I was also supported financially to bring a caregiver along to do a research visit, which is not actually that common in New Zealand, but something that really helped me. And I also spent a lot of time reassessing my priorities and dropping some of the service roles that I just really couldn't manage while I was working part-time. So I've written a little bit about this in my blog, so you're welcome to go along and check that out if you're interested. And I'd be happy to talk to anyone about that if you'd like to at any time as well. So I'll just finish by thanking the very many collaborators, students, field assistants, mentors, funding bodies that have all supported my research, and particularly to my village. So I'd like to thank my husband in particular for doing a huge share of the parenting, and that's really allowed me to chase my academic dreams and take on this craziness that is a faculty job. And I'd also like to make special mention to Greg Holwell, who's been my long-term collaborator now, and who was my PI when my son was born, and a large reason why I was able to transition back to work so smoothly, smoothly as it could be during this period. So thank you a lot to Greg. And if there's time, I'd be very happy to take questions. And kia ora koutou.
Oh, thanks, Chris. That was fantastic. I think I was just going to look and see how much time we've got. And people, if they've got questions, can go ahead and raise their hands. While we're waiting for other people to raise their hands, I'm going to just jump in and ask about this whole like condition dependent thing with a lot of these weapons, and ask about like with dung beetles, sometimes the females will, like the size of the dung ball they give them will affect whether or not the males get big enough to develop these weapons and stuff. Do we know anything about this species or anything about like egg size for females, or how they could set their eggs up to be in a better position for being a major male?
Yeah, that's a really good question. The short answer is no, we don't know, but it's very likely that the heritable component of being big is only a small part of the story, and it's probably a lot partly to do with how big the egg is to start with, but also where she chooses to place it. So females, oh man, as I've discovered this summer by trying to track females instead of males for a change, they're incredibly picky about where they lay their egg, and they're often aggregating in really, really tight clumps together. So there's obviously some cues that they're picking up about the trees that they're laying into that make it a good spot for their babies. But I'd say that it's largely to do with the nutrition that they get from the log that determines their size, as well as some heritable component as well.
It's really cool. Sounds like lots still to do with that whole career on the brantids. Does anyone else? We only have a few minutes here, but if anyone's got some other questions, it looks like Jeff is raising his hand, but he's not doing it in the appropriate way, so he's lucky I saw him. Because
I'm one of the hosts, I don't have the option.
Okay, fine. You can have a question then.
All right, thanks. This is going to be kind of a facetious question, but also kind of serious. Has anyone done a kind of a weevil or harvestman fight club kind of thing, just to see if there's, is there a rock, paper, scissors kind of thing going on with these different polymorphisms?
Yep, that's a really, really good question, Jeff. So with the weevils, I guess it's a bit more simple because they just vary along kind of this linear relationship between small and large. But for the harvestmen, that's absolutely a really good way to kind of figure out, I guess, whether in some situations it's beneficial to be long and slender versus some situations where it might be better to be short and broad. The short answer is we've kind of tried. It turns out, even though these are super awesome species, they're really difficult. The harvestmen are really difficult to work on. But I think that's quite likely to be what might be maintaining the kind of ratios of the different morphs within the field, because you certainly see that oftentimes the really long slender ones are a lot more common than the short broad ones. So that kind of does suggest that there might be some frequency dependent kind of selection going on there. But yeah, it would be really great to be able to do some fight clubs to kind of look at that for sure.
Anyone else want to wave their arms around or pop up their hands in the participants box? Mostly I'm just in the chat. You're just getting loads of love, both for your triumphs and your struggles. And also I think you've convinced everyone that giraffe weevils are like a fantastic species that people should get hopped up on.
That was my only goal for the day. So
how common do we have a bunch of species like that in New Zealand of the weevils, or is it kind of like our flagship weevil?
No, it's like lots of, I guess, groups within New Zealand, we've only got the one representative. So it's a little bit frustrating because I guess it would be really nice to be able to do some comparative behavioural work within New Zealand. But there are a lot of brentids in Australia. So I think that's kind of the next frontier for doing brentid work behaviourally. So especially in northern Queensland and the tropics there's lots of species with these really exaggerated weapons. And then as you move down the country into, I guess, temperate New South Wales and Victoria, the species are much smaller and they're actually almost, I think, all associated with ant nests. So they're inquiline species, and nobody knows what they're up to at all. So yeah, lots of opportunity to do some cool comparative stuff across the east coast of Australia, I think.
Super cool. We got time for like one more question. I think Anna's got one. So go ahead, Anna.
Hi, Chrissie, such an amazing talk. My favorite part was the sneaky copulators. How do they get in there and what makes them successful and the bigger males not great at getting rid of them?
Yeah, that's a great question, Anna. So I guess a lot of the time, the little ones just try and avoid touching the big male at all, so they're not even detected. I don't think their eyesight's very good. And it's kind of funny, like there's lots of jokes you can make about them, right? Because they're often mating under the nose of these larger weevils kind of thing. So they literally often don't see them. And when they do see them and detect these sneaker males, then they, as you might have seen in that video, they kind of tuck themselves down the side of the female. And that reduces any area basically that they can get pulled off, despite a lot of best efforts. But you do see a lot of sneakers getting thrown off the tree too. So it's not an entirely safe maneuver.
So in weevils that I know, which are your lovely giraffe ones, they're like really like not sticky legs but grabby legs. Is that what they use? Is that what these guys use? Like hooks? Like
yeah, they have kind of sharp little claws on their tarsi that